Hello, in this video we're going to deal with the Nobel Prize in Physiology or Medicine from 1910, awarded to Albrecht Kossel in recognition of the contributions to our knowledge of the cell chemistry made through his work on proteins, including the nucleic substances. First, some information about his work coming from the official Nobel Prize site. Here you see the link. The fundamental process of life takes place in cells of organisms. A variety of chemical substances are involved in an equally rich set of processes. From the 1880s, Albert Kossel studied the biologically important substances, proteins, DNA, and RNA, and their components. Among the things he investigated protamines and histones, proteins that together with DNA form complexes that build up the chromosomes in the cell nucleus. This knowledge later became important for the development of genetics. Then we continue with some biographical information also coming from the Nobel Prize site, which is summarized by me. Kossel's field of work was physiological chemistry, especially the chemistry of tissues and cells. His activity as a teacher in the university, however, extended to general physiology, which in his time was in most German universities still not separated from physiological chemistry. He began his investigations into the constitution of the cell nucleus at the end of the 70s, and in the 90s he turned more and more to the study of the proteins, the alterations in proteins during transformation into peptone, the effects of phenol diet on urine, the peptonic components of the cell, the simplest proteins, etc. Working on fish roe, he studied the protamines and hexosome bases. In 1896 he discovered histidine, then worked out the classical method for the quantitative separation of the hexosome bases. And with the distinguished English pupil H.D. Dakin, he investigated arginase, the ferment which hydrolyzes arginine into urea and ornithine, and later he discovered amytine in herring roe and devised a method for preparing it. Among his important publications may be mentioned Untersuchungen über die Nukleine und ihre Spaltungsprodukte, that's German, investigations into the nucleons and their cleavage products. In 1881, Die Gewebe des menschlichen Körpers und ihre mikroskopische Untersuchung, Tissue in the Human Body and the Microscopic Investigation, which is in 2 volumes, written between 1889 and 1891. And it was written with Birons and Schierdecker, en de Leitfaden für medizinisch-chemische Kurse, textbook for medical chemical courses, 1888. Since reprinted several times. He was also the author of Die Probleme der Biochemie, of the Problems of Biochemistry, from 1908. Die Beziehungen der Chemie zur Physiologie, the relationship between chemistry and physiology, which was a contribution to Kultur der Gegenwart, the culture of today, from 1913. As said, it's summarized by me. If you want to read the complete biography on the Nobel Prize site, follow this link. Here you see some of the compounds he investigated, in this case pyrimidines, cytosine, thymine, and uracil, which are in the DNA or RNA, as you can see. And here you see how they form base pairings. Adenine and uracil combine just as guanine and cytosine. Okay. Here you see some more pyrimidine nucleobases which can be modified into other compounds, coming from the Wikipedia, as you can see. Then the award ceremony speech by Professor the Count Mörner, also summarized by me. Many vital phenomena are of course such that chemical research, at least at present, is far from being able to eliminate them to any real extent. This is the case, for instance, with mental activity and the manifestations of life related to it. This is 1910. Other vital phenomena are more easily accessible to the chemist and have for long been the object of his work. This applies to such objects as nutrition, growth, metabolism, and the chemical composition of the organs and body fluids. The working methods chosen differ according to the angle from which the problem is attacked. in, for example, the study of metabolism, which has been carried out for several decades, the amounts and type of the nutrients which are required by the organism under different conditions are established, and the metabolic end products which are excreted by the organism are determined. Such a determination of the intake and output of the organism gives a picture of its overall economy and can also give certain information on internal factors of its management. The problems are attacked in a more direct way by endeavoring by direct experiment to determine the composition of the different organs, their functions, etc. In this, the effort of the anatomist, the histologist, the experimental physiologist, and the chemist go hand in hand as they seek together to penetrate the dark secrets of life. The task of the chemist is to determine the chemical structure of the parts of the body and the chemical process taking place within them. The study of living organisms has more and more led to the view that its smallest independent units, morphologically speaking the cells, are also to a certain degree lead an independent life and are the real seeds of the vital processes. The cells therefore attract special attention in biological research and studies which widen our knowledge of the cells to any important extent deserve to be given prominence. The cells of the organism exhibit on one hand certain special characteristics according to the organs to which they belong, and on the other hand general shared characteristics as well. It has long been known, and it's easy to demonstrate, that the main masses of their organic substance consist of materials belonging to the group of protein bodies. They are, however, dissimilar in different cases. The development of the protein chemistry in general, and of the specialized knowledge of the protein bodies occurring in cells and of the combined forms in which they appear, is therefore an important link in the chain of knowledge to cell chemistry, and consequently also a factor of great importance in research on cellular life and functions. Professor Kossel has made important contributions to research in the ways we have just mentioned. This is not the place to give a detailed account of this work, but I cannot leave trying in a few words to indicate its significance. For some decades, highly successful attempts have been made to learn more of the detailed structure of these biological significant proteins by studying their breakdown products. It has been shown that proteins are usually built up of a large number of mutually differing atom complexes. These can, by and large, be arranged in two major groups, the mono amino acids and the basic breakdown products. Among the many workers who have studied the first group, the Nobel winner for chemistry in 1902, Emil Fischer, takes first place through this brilliant analytical and synthetic studies. The second group of the breakdown products is nevertheless no less important, and in the investigations of these, Professor Kossel has earned great credit. The first really clear picture of these substances was given by Drexel and his student, among whom the name of Swedish research worker who has recently returned to his native land may be specially mentioned. Professor Kossel has since then enlarged the knowledge of the discovery of new related substances in particularly he has, by means of extensive work using new methods which he himself elaborated, carried the knowledge of the quantitative relationship of these substances in the protein molecule further than is the case for the other protein breakdown products. There are several kinds of proteins. One group which is included here are the so-called protamines obtained from the milt of fish. Kossel has made a detailed study of these. For these, a relatively simple structure has been discovered inasmuch as the number of dissimilar atom groups in them is not very great. They therefore present simpler relationships than proteins in general, and consist mainly of substances belonging to the group which I have just called basic breakdown products of protein. For certain protamines, Kossel, thanks to his method of determination, has in fact been able to establish the quantitative relationships of the building blocks making up these protamines, a goal which we seem to be far from attaining, where the other proteins are concerned. Work on these most simple protein bodies, that is the protamines, is however not only of great importance because it has explained the structure of such protein bodies. The protamines are also of direct interest for the knowledge of certain cells and their life because they are in fact characteristic of certain transformation products of the cells and are formed from ordinary protein. One protein group, first observed by Kossel, consists of the so-called histones. They stand between the protamines and what is termed ordinary protein. This group again is important because of its occurrence as a component of certain cells and has also been studied in detail by Kossel. Professor Kossel has made an extensive and important study of the problem of the protein compounds in cells. As we have already mentioned, the proteins are a very complex body. Within the cells of the relationships are further complicated by the fact that the protein cells are combined in various degrees with other substances, such as those grouped under the name of nucleic acids. These two are far from simple in structure. They form phosphorus-containing organic substances which, together with a form of phosphoric acid, usually contain purine bodies, pyrimidine bodies, and a form of sugar or related substances. I will restrict myself to remarking that these nucleic acids, by their presence in the cell and their relation to the protein bodies found there, certainly possess a great biological significance, and furthermore I should like to emphasize that it is to Kossel and his pupils especially that we owe our present advanced knowledge of these problems. Okay, this is from the award ceremony speech, summarized by me. If you want to read the complete speech, follow this link. Here we see a compound group mentioned in the text, the purines. Important members adenine and guanine are also present in RNA and DNA. I think, no, yeah, that's true. Here you see some of the modified versions of these two compounds coming from Wikipedia. And that was it for the moment. Next week we will go again into this Nobel Prize, and then we will I will give you a summary of the Nobel Prize lecture given by Albrecht Kossel. For now, thank you for watching, and maybe till another time.